Viruses are the most abundant biological entities on Earth, outnumbering all cellular life by orders of magnitude. They sit at the edge of what we call life, yet they shape evolution, drive global biogeochemical cycles, and, at times, rewrite human history. Today, we'll explore mysterious viruses that could change our reality. Ancient pathogens thawing from permafrost, giant viruses that blur the definition of life, emerging threats amplified by climate change, and engineered viral systems poised to transform medicine, alongside the ethics and biosafety questions they raise. First, a quick primer. A virus is a genetic package, DNA or RNA, inside a protein shell, sometimes wrapped by a membrane. Without a host cell, it cannot reproduce. But that simplicity hides astonishing diversity. Viral genomes range from a few thousand bases to millions. Some viruses hijack cellular machinery with ruthless efficiency. Others carry toolkits, DNA repair enzymes, translation helpers, that look more like the gear of living cells than the minimal parts of a parasite. This continuum challenges our binary categories of living versus non-living, and forces new thinking about the origins and evolution of life. 1/4 of the northern hemisphere overlies permafrost, frozen ground that has preserved microorganisms for tens of thousands of years. As Arctic regions warm roughly 4 times faster than the global average, this biological archive is thawing. Researchers have revived multiple eukaryotic viruses from Siberian permafrost, some approximately 48,500 years old, spanning families such as Pandora virus, Cedrit virus, megavirus, Pac-Man virus, and pathovirus. Remarkably, Pathovirus Sibiricum, up to 1.5 micrometers long, retained infectivity after approximately 30,000 years. These findings demonstrate that frozen environments can preserve viral viability across geological timescales, and that abrupt thaw can co-release microorganisms from different eras, complicating risk assessment. A 2016 anthrax outbreak linked to a thawed reindeer carcass shows that release of ancient pathogens is not merely theoretical. The 2003 discovery of mimivirus overturned the notion that viruses are always simple and small. Giant viruses can have genomes larger than some bacteria and encode proteins associated with metabolism and DNA repair, features once thought exclusive to cellular life. Pandora virus salinus carries approximately 2.47 million base pairs. Some scientists have even proposed these giants as candidates for a fourth domain of life, a provocation that underscores how incompletely we understand the viral tree. Whether or not that claim holds, giant viruses expand the biochemical repertoire available to evolution and to biotechnology. Isolated from a Brazilian lake, Yara virus is startling for another reason. 90% of its genes do not match any known sequences. That means the majority of its genome represents previously unseen biology. Virologists note that more than 95% of viral sequences in environments like sewage also lack matches in reference databases. In other words, what we know about the virosphere is likely the tip of an iceberg, and most of it remains unmapped. Discoveries like Yara virus are less a conclusion than an invitation to look deeper. Medusa virus adds yet another twist. Rather than producing complete progeny inside the host, it assembles components that are released and later recombined, more like modular Lego-style assembly than a single factory line. Infected amoebae harden before lysing, a pathology consistent with the virus's myth-inspired name. Mechanistic outliers like Medusa virus broaden our models of how information and matter flow during viral replication, and suggest there may be many uncharted strategies in the wild. Climate hazards are already known to aggravate a majority of human infectious diseases. Warming and altered rainfall expand the ranges of vectors, mosquitoes, ticks, while heat acts as a selection pressure favoring heat-tolerant viruses better able to withstand fever. Oropouche virus, historically Amazonian, has shown genetic shifts that enhance replication and transmission, with thousands of cases reported across several countries and travel-linked detections abroad. As ecosystems shift, so do viral host and vector networks, increasing the opportunity for spillover into humans and livestock. Surveillance and vector control, long underfunded, become essential climate adaptation tools. Highly pathogenic avian influenza H5N1 has recently infected U.S. dairy cattle, raising exposure risks for workers. In 2024, dozens of human infections were documented in the United States, primarily in occupational settings. Historically, H5N1 case fatality has been around 30%, And concerningly, the virus's adaptation to mammals has not been accompanied by clear attenuation in virulence. While sustained human-to-human transmission has not been observed, the combination of mammalian adaptation, high pathogenicity, and widespread animal reservoirs define a risk landscape that warrants aggressive surveillance, vaccination of at-risk animal populations where feasible, and robust occupational protections. Not all viral futures are threats. Synthetic biology is turning viruses into precision tools. Researchers have built protein-based logic circuits using engineered viral proteases that execute and/or/not operations inside living cells. In oncology, such circuits can be designed to sense multiple cancer biomarkers and then trigger therapy only when the logic matches a tumor signature, reducing off-target effects. This is computation performed with proteins in vivo, enabling smart therapeutics that adapt to the cellular context. Virus-like particles, VLPs, mimic the shells of viruses but lack genetic material, eliminating replication risk while preserving immune stimulation and delivery capacity. VLPs already underpin licensed vaccines, and their modularity allows decoration with antigens or cargoes for targeted delivery, even construction of artificial organelles. Meanwhile, adeno-associated virus AAV vectors have transformed gene therapy, with approved treatments for inherited blindness and spinal muscular atrophy. Next-generation capsids engineered for specific tissues and lower immunogenicity promise broader indications and repeat dosing, long-standing challenges in gene therapy. With power comes responsibility. Gain-of-function experiments can illuminate how viruses evolve, but also carry dual-use risks. History shows that even high-containment labs can fail. With more than 1,500 BSL-3-4 facilities globally, small per-lab risks accumulate to a meaningful global probability of accidental release. Best practices: transparent risk-benefit review, stringent biosafety, biocontainment, international reporting norms, and robust training are essential to keep research benefits while minimizing catastrophic downside. AI is accelerating virology. Tens of thousands of new viral species have been identified computationally from environmental data. Machine learning now helps predict host range, antigenicity, and immune escape, and can propose designs for vaccines and vectors. On the basic science frontier, unusual entities such as minimal symbionts that verge on viral lifestyles force us to rethink the lower bound of what counts as a cell. In parallel, the viral enzymiome within giant viruses may be a biotechnology goldmine, novel catalysts for synthesis, DNA, RNA processing, and beyond. Viruses are both an existential risk and an extraordinary opportunity. Permafrost awakenings, climate-driven spillovers, and avian influenza adaptation underscore the need for vigilance. At the same time, programmable circuits, VLPs, and advanced vectors point to a therapeutic future where viral design saves lives. The path we choose, investing in surveillance, integrating climate and health policy, enforcing biosafety, and funding trustworthy, transparent research will determine whether these mysterious viral forces become harbingers of crisis or catalysts for a safer, healthier world.